as we go along. Dr. Amina, you'll give me the cue when to begin.
Oh, sorry, I thought I was speaking actually. Ladies and gentlemen, welcome to today's session. We're going to discuss about approach to a patient with a genetic disorder by Dr. Sukwao Ilovi, who's a clinical geneticist. And this is, like a new evolving field in medicine. So we're going to have this discussion, and after that we'll have the Q&A. Kindly remember, you can post your questions on the Q&A. It's open for everybody to post. And as usual, we'll have this updated and uploaded on our KNH YouTube channel. And follow us on Twitter. It's a new handle where we'll keep you updated on most of our activities. So Daktari, it's a real honor and pleasure to have you here. And welcome. We can't wait to learn more from you.
Thank you, Dr. Amina, for the kind introduction. It's also a pleasure for me to be able to speak to you this evening. So as Dr. Amina has introduced me, my name is Dr. Sukwao Ilovi. I'm a clinical geneticist and lecturer with the University of Nairobi. My slides are not moving. Okay. So the objective of this lecture is that we should be able to have a systematic way of approaching a patient. So we'll revisit this case at the end of the class. So I'm going to read it out. We put it at the back of our head. Then as we are going through how to approach a patient with a genetic disorder, we will discuss it. So we have, I took a verbatim report as appeared in a newspaper. So typically how patients will present to us. So clinicians at a university hospital first encountered the aggressive family when several women of childbearing age sought genetic counseling in 1978. Certain family members, always male, were mildly mentally retarded. More disturbingly, the male members of the family were prone to aggressive, impulsive outbursts during which they would threaten and fight with people. The violence streak had a long history, and in 1962, the woman's grand-uncle had actually prepared a family tree which had identified other members of the family afflicted with the same disorder, tracing it as far back as 1870. So this is a disease that has been in this family for 100 years. So we'll revisit this. And genetic diseases, some fall in the realm of common genetic disorders like sickle cell, cystic fibrosis. Some fall in the rare disorders, so that a patient might present to you. You don't know what you're dealing with, but at the end of the day, you have to formulate some roadmap to be able to begin to answer the question. So who are you genetically? At the end of the day, we are just a series of nucleic acids, pretty much like a computer code, which are encoded in your DNA. Your DNA is housed within your chromosomes in the nucleus and mitochondrial DNA. So this sequence codes for certain things, and that is what we call a gene. So maybe line 1 is coding for a gene like insulin, line 2 is coding for melanin, line 3 is coding for osteoid protein, so that your body has an instruction, a genetic code that tells it, with this code, we will produce a certain protein which has a certain function. And so that genetic disorders begin to develop when you deviate from that. So that if this is a code for a certain protein, just a simple substitution, maybe the T changes to a C. Then it changes the nucleic acid and it changes the amino acid sequence and protein so that you can begin to have genetic disorder. So essentially at the end of the day, we are just a series of C, T, A, and G. And how are we different from other animals, plants, and fungi? Human beings, all human beings on Earth, their nucleic acid sequence is identical 99.9%. We have 3 billion base pairs. So 99.9% of that is identical. So it's only the 0.1 that gives us the difference in how we look, how we behave, and so forth. When we come to other primates, it drops down slightly. When you come to something like a banana, we actually share 60% of DNA. So that in the regions where we share, like if this is the region where it is identical to a banana, if I looked at this nucleic acid sequence, I would not be able to tell you whether this has come from a banana or a Homo sapien. So how is this possible? It all originates from having a common ancestor. So life on Earth began 3.5 billion years ago, and it began with a single organism that was able to replicate asexually, so that all living organisms can trace their ancestry to that single cell organism 3.5 billion years ago. Then it developed, divided asexually, formed two. So as it starts to deviate, it starts to have mutations, so that you see as you go along the way, maybe 2 billion years ago, we branched off from animals, so that in the animal kingdom, our closest relatives are actually fungi. So that if you look at you and your brother, you and your brother have identical ancestry. Your most recent ancestor are your parents. But behind your parents, you have shared common ancestry all the way to 3.5 billion years ago. If you look at you and your first degree cousin, you don't share parents, but you share grandparents. So your most recent common ancestor are your grandparents. Then after that, it becomes the same family tree. So the more you look alike, the more recent your ancestry can be traced to a common ancestor. And the more different you look, the further back you have to go. So how common are genetic disorders? I want to give you two autosomal recessive disorders, which means you need to have inherited the mutated gene from mom and another mutated gene from dad. So how many of you seen a child with spinal muscular atrophy or albinism? They are pretty rare, but if you sit down and think, you'll say, yes, actually, I've seen one maybe in the last 10 years. So they are not that common. That is for me who practices adult medicine. But if you talk to people, even who are practicing pediatrics and maternal fetal medicine, they are not that common. They seem to be rare. So are they really rare? So the first one we discussed was spinal muscular atrophy. If you find a child with SMA, what you are witnessing is an occurrence that occurs once in every 10,000 births. But then if we go and look at the carrier status, it's actually 1 in 50. So that of parents who are carriers, so they are recessive carriers of the gene, when they procreate, the likelihood that they will have a homozygous affected child is actually only 1 in 4. The other 75% of the time, the child phenotypically becomes normal. So that if you're going to mathematically calculate backwards, the actually carrier state of spinal muscular atrophy is 1 in 50, and it's been found from population studies. When you look at live birth rates, the live birth rates incidence of spinal muscular atrophy is 1 in 10,000. Carrier states within the population is 1 in 50, and this transcends various races and ethnicities. So what does that mean? It means 1 in 50 carries the gene. We are 200. So it means at least 4 of us have this spinal muscular atrophy gene. And in most instances, it doesn't cause a problem, because if that person doesn't meet with somebody who carries the mutated gene, their children will never be affected. And even in the event if they pair up with somebody with the gene, 75% of the time, they will not have any problems. So now we look at the other one. Albinism occurs at an incidence of 1 in 18,000. If you think back, it's pretty rare. You don't see it that often. But if you look at the carrier state, if you work it backwards mathematically, it's 1 in 68. And population studies have shown. So that being carriers of genetic disorders or potentially genetic disorders, which are what we call silent, is pretty common within us. So there are over 6,000 known genetic disorders. And these are the genetic disorders where they have mapped the genotype. If we include the ones that are suspected to be genetic, but people don't know exactly what causes them, the number rises up to almost 8,000. And genetic disorders can be broken down into 3 monogenic, meaning one gene is responsible for the mutation. So like what you've seen for spinal muscular atrophy, for sickle cell disease, hypertrophic cardiomyopathy, hemophilia, autism spectrum disorder. Then we have complex trait disease. Yes, there seems to be a strong genetic composition, but it's not so clear cut. It looks like such a number of genes are acting in concert, so that like diabetes, there's been about several hundred genes that have been found to be, so that it's not very clear how they seem to run in families, but you're not able to predict how they're running. Then we have chromosomal disorders, so that a disease like trisomy 21 occurs in 1 in 500 or 1 in 600 parts. So it's pretty common. A disorder like Turner syndrome, which is also a chromosomal disorder, occurs in about 1 in 2,000 parts. So they are not particularly rare if you actually break them down. So individually, we all carry about 10 to 15 genes that are potentially of concern. Most of us might go through life never having to deal with the consequences of those, either in ourselves or offspring, particularly if they are recessive traits. Sometimes they manifest, sometimes they don't. So that it's on average 1 in every person carries as a recessive trait. The rate for hypertrophic cardiomyopathy is 1 in 500, and we've already said Down syndrome occurs at a rate of 1 in 600. KNH delivers slightly less than 1,000 births per month. So it means every month in Kenyatta, maybe 1 or 2 children will have Down syndrome. So they're not particularly rare disorders. And then when we're looking at the phenotype, we know there's a genotype behind it. There's a certain deviation from the normal genetic sequence that has led to the disorder. So that a disease like polycystic kidney disease, where we know it's exclusively genetic in origin, you might do genetic tests, and in 15% of the time of the patients, you actually don't find the genotype. So what you tell that family is that, yes, you clearly have a genetic mutation based on the phenotype and based on the pedigree, but as of now, there is no test. Maybe it's a new mutation that is in your population, that's or in your family, that it has not yet been discovered. And this becomes one of the difficult things of trying to provide genetic care, because sometimes you do very expensive tests. They can range anywhere from 50,000 to 500,000. And at the end of the day, you're telling people, I don't know what has caused the disorder. So this is a very interesting study. It was looking at prevalence of chromosomal abnormalities on just karyotype, on sperm donors. You know, the entry point in a French sperm donors, you know, the entry point for sperm donors tend to be pretty stringent. So the French, they require somebody to actually have delivered a child, and the child to be healthy for you to qualify or to become a sperm donor. So usually with the history, you try and find out at least no first degree relatives. First degree relatives is the sperm donor, parents, sibling, and children. So essentially people who are relatively well of above average IQ, doing well. So they went and found actually they had abnormal karyotypes in 38 of the 10,000. So they are not, so these are what you are calling asymptomatic disease. They might not cause problems in the sperm donor, but sometimes they go and cause disorders in the subsequent generations. And a karyotype is done by looking at chromosomes under a light microscope. So what you're looking at is genetic material, what essentially is gross or macroscopically. If we go to the sequence level, this 0.38% certainly would be much higher. So even apparently normal healthy men who qualify for sperm donors seem to have quite a number of abnormalities, even just on karyotype. So what is the role of a geneticist? My role as a geneticist is to provide transgenerational, largely diagnostic services running across many generations. It's usually what you're seeing, the phenotypes tend to be just a tip of the iceberg. What can look very simple ends up being very complex if you go to the genetic level. Sometimes even the phenotype is so complex, you're trying to figure it out. Sometimes we don't know what you're dealing with, and essentially we go on a hunting expedition and try and catch as many fish and try and see that will give us an answer. This is a pedigree, and this demonstrates some of the challenges that we have. So this is a female child who presented with what was autism and intellectual disability. And they did a gene panel looking for disorders that are gene mutations that are associated both with autism spectrum disorder, intellectual disability. And this mutation is highly specific for autism. It's inherited as an autosomal dominant trait. So they went and tested the father. The father has the same mutation, but is of normal neurological function. So when this couple wants to have another child, and they're asking, will the other child be affected? So first you say it's an autosomal dominant disorder. The likelihood of a future child inheriting is 50%, but I cannot tell you where in the spectrum. They might be completely normal like that, or they might be affected like the sister or even one. So it becomes sometimes impossible to gauge what the outcome will do, so that it's almost a wait and see approach. So reproductive genetics tends to be the most complex because sometimes you're not able to give people definitive answers. So the basis when you're talking about genes, you're going to the nucleic level. So chromosomes are located in the nucleus. Mitochondria are located in the cytoplasm. So those are the two sources of our DNA material. And we inherit 22 autosomes, one from mom, one from dad, and sex chromosomes. And then mitochondrial is exclusively inherited from females. So this is essentially what a karyotype is. The chromosomes are arranged in size so that 1 is the largest all the way down to 22. They're in decreasing length. The only one that is larger, with the exception of 22 being larger than 21. So how a karyotype is performed, we'll talk about it later, is actually looking at this, how your patient's chromosomal material deviates from the known normal karyotype. And when you open up a chromosome, you can see your nucleic acid sequence, your T, which is pairing with A, your C pairing with G. All the way 6 billion base pairs if you count from chromosome 1 all the way to your sex chromosomes and mitochondria. So what a gene is the length of nucleic acid that codes for a protein. And these genes have been mapped. 95% of human genome has been mapped. So let's just look at chromosome 11. It has been mapped. It's been found to have about 2,000 genes. It's has 130 million base pairs, of which 95% have been determined. So the 5%, this 5% that seems to be undetermined, sometimes might explain why sometimes we do genetic tests on patients and we're not able to give them answers. So we know at what end we have the gene that codes for insulin. And this is the nucleic acid sequence for insulin. It begins all the way there. So that this is what is the reference sample which you will be comparing. If you're looking an abnormal gene sequence, you'll be comparing it with the known normal reference sequence. Now the challenge comes in is a lot of these databases are using Eurocentric reference sequence to look different from Europeans. So it means even our normal will be different from Europeans. So that the challenge we run with genetic tests, when we're running them against reference sequences of people who have a genetic makeup that is different from yours, you run the risk of not being able to answer some of the questions you are getting. And genetic material needs to be just the right amount. If you have too much, you end up with disorders like trisomy 21, which is Down syndrome. If you have too little, like of sex chromosomes, you end up with Turner syndrome. So everything in genetics just needs to be just right. And your genotype is what dictates your phenotype. Your C, T, A, and G in the DNA is what will determine how you look. So that from there you have your DNA sequence, you get it transcribed and into an mRNA, into a protein. And you can do tests at any of this level. You can test at the gene level, you can test at the mRNA level, you can test at the protein level. Sometimes you can even test at the metabolite level. And so that if you're looking at a test like sickle cell, we usually measure hemoglobin S, which is the protein, right? And that is able to tell us what is going on upstream And that will also tell us what is going on downstream, which is the phenotype that with hemoglobin SS, we expect to have the phenotype, which is called red blood cells. So the concept of gene expression, at the early stages of the zygote, you have pluripotent stem cells. Every cell in the human body contains all these chromosomes, all 23 chromosomes and mitochondrial DNA, with the exception of red blood cells, which neither contain a nucleus or mitochondria. So that all these are expressed in the pluripotent cell. A cell is able to express and give you all sorts of things, from your protein, your retina, pigment, your melanin. Once it becomes committed, once it moves from pluripotent stem cell, pluripotent cell to committed, then it switches off, so that your retina has all these genes, but it will only express. Express means it will only produce an mRNA sequence, it will produce a protein sequence that is relevant to the retina. Your pancreas similarly has all of this, but it will only produce, when you're talking about your beta Langerhans cells, they will only produce one tip of chromosome 11, which is with insulin, and so on and so forth. So that the reason why pluripotent cells, if you look, okay, it's unethical to use fetal tissue in most instances, but why they create quite a bit of excitement is that you are able to make a cell do just pretty much anything, so long as you can gene edit, make it do 1, 2, 3, and that you can generate from that pluripotent cell. You can say, I want to generate both, you generate. I want to generate a pancreas, you can generate. So that is the concept of gene expression. Despite every cell having every gene, it has been programmed and committed to only produce cells that are relevant to that system. So where do our genes come from? We can either inherit, and you can inherit benign or pathogenic variants, or they can occur de novo, and those also can be benign or pathogenic. So now we come to the modes of inheritance. If you're inheriting genes, if it's a single gene, they can be inherited in a Mendelian inheritance, which is usually as distinct units in a predictable manner. So autosomal, X-linked, Y-linked, dominant, recessive. And this is what gives you your single cell disorders, from sickle cell, Huntington's, some inheritable cancers, autism spectrum disorder. So that if I'm looking at the gene level, a patient who has sickle cell, I know exactly which gene to go and look at. I will go for that gene, because there's only one gene mutation there's only one gene that gives you sickle cell. If you look at something like hypertrophic cardiomyopathy, there are about 4 or 5 genes that have been found to be associated. So I'll do what is called a gene panel for hypertrophic cardiomyopathy, which will have those 5 to 8 genes to try and establish which one is the mutation. So non-Mendelian inheritance are the ones that had not fall in the previous category. So that, like what we talked about, diabetes, hypertension, some asthma, cancers, and the like. So I want us to mention a bit about autism spectrum disorder, because this is a disorder that is increasing. It's largely genetic because it's static. Someone is born with it. So something clearly happened either uteral or inherited. So it has both monogenic forms, and in one family, you can have somebody who's completely asymptomatic, somebody who has, another person has intellectual disability, somebody else has poor social interaction, somebody else has ADHD-like syndrome. So that it doesn't all, even within the same families, they can have different expressions. So that this is a monogenic form of autism spectrum disorder. So the father was found, and then all his children, 3 children, have. And so we know this is inherited in an autosomal dominant way because they have the phenotype. And you know, fathers transmit autosomes and Y chromosome to their sons. Fathers transmit autosome and X chromosome to their daughters. So we know now here the common denominator is autosomal, because this has allowed us to rule out both X and Y chromosomes. And then now this is a polygenic pedigree where you can't quite tell what is it's not so clear cut what is the mode of inheritance. So how do we evaluate patients who seem to have a genetic disorder? So there are certain entry points for a genetic evaluation. So a patient who's known to have what is clearly a genetic disorder. So polycystic kidney disease, hypertrophic cardiomyopathy, somebody who's suspected to have a genetic disorder, so that a child who gets a sudden cardiac death is highly unusual. So it's almost certainly a genetic disorder. We need to start thinking in that way. Or a 15-year-old girl coming down with CA ovary, we know that is not the age group. So that should prompt us to think, is this an inherited cancer syndrome predisposing them to get it? CA ovary at such a young age. And this thing of diseases that just run in the family. That's patients come to you and tell you there's something that runs in the family. Like the case we had started with. And in anything in genetics, when you have a history, the next point is a family history, which is a pedigree. Pedigree is what an ECG is to a cardiologist, it's what is a pathogram to an obstetrician. It tells us everything. I don't even have to have a history, and we'll go through it so that just with the pedigree can tell you quite a bit. We've had a few pedigrees as you're going along, and you can begin to see it gives you an idea. So ideally, you should have 3 generations, the affected person, their siblings, and first-degree cousins. That's one generation. Then the generation above, which is their parents and their parents' siblings. Then you can go another generation up, the grandparents, or even one down. So as many as you can get. The more you get in the pedigree, the more informative it is that it begins to give you answers. And by and large, I'll keep it short. There are signs. Square denotes male, circle female. If they are joined with a horizontal line, that is a relationship line. Consanguinity usually has 2 lines, one which is a relationship line, another one is a bloodline. Then now this one tells you that this, broke down. If there is a death, it's crossed out like this one, like so that this one was a miscarriage, so it's a death. If it's shaded, something like that. Okay. So I'd like to mention something about consanguinity. Consanguinity is a time-honored tradition that has existed for millennia. It's currently practiced in 1 billionth of the world's population. It has a lot of social and cultural benefits. So like there's one huge genetic study, which is called Born in Bradford study. So they looked at women presenting to antenatal clinic, and they asked them questions. They grouped people who are in consanguineous unions and non-consanguineous unions. And there are certain benefits, so that the people who are in consanguineous unions, when the women are less likely to smoke, they are less likely to have not paid their utility bills and previous month's rent. And that is largely because, the communities that practice consanguinity also practice extended family living, which is tends to be better for mom and baby. So how you assess consanguinity, you have said it's 2 lines. And why it's important, so that like in this family, these are first-degree cousins, because they don't share parents, they share grandparents. So these are first-degree cousins. So their grandmother had a recessive trait. A recessive trait can survive centuries in a family, so long as it doesn't meet another recessive trait. So what happened? Grandma passed it on to her daughter on this side and her son. Then when they had children and they got married, you have the same gene being present in both of the parents. So what consanguinity for geneticists is of concern is that it tends to bring out autosomal recessive disorders in a higher frequency than expected in a non-consanguineous relationships. And when we're talking about consanguinity, we are talking about people who share 3.125% of their DNA, second-degree cousins and closer. Why it's cut off at second-degree cousins and closer? It's been shown any other unions that are further down, the rate of autosomal recessive traits is not higher than in the general population. So I'd like to highlight a case we saw in Kenyatta, a patient who presented with an exceptionally rare form of renal tubular acidosis, which has been reported to occur in about 1 in 80 million births. And that gives us a carrier rate of 1 in almost 4,500. So it's pretty rare. So what are the odds that 2 people walking around who are non-related are carriers of this gene. And when we ask them, actually they were second-degree cousins. So that this gene that had gone down the family tree 2 ways is actually the same gene. And consanguinity is of concern the rarer the genetic disorder. A disease like sickle cell disease, which has a carrier state in malaria endemic areas of Kenya of up to 1 in 31, is not of concern. It not occur in a higher frequency in consanguineous unions compared to others. So how do we interpret a pedigree? So this is a case we published. We took the genetic history from the relatives. They usually require 2 sittings. One to try and map out, and then another one they need to sit back and possibly call. It's usually an auntie who is the custodian of the family history. So it usually takes 2 sittings. If an inpatient, I will tell them I'll probably come later in the day or tomorrow. If it's an outpatient, you can tell them, is there somebody you can call? You can sit outside for 30, 40 minutes as you call around and then come in with this information. So there are certain things we can get. Usually the proband, sorry, it's not showing. The proband, our index patients is denoted with an arrow. So it's this 61-year-old male who presented to our inpatient who was known to have polycystic kidney disease on ultrasound and end-stage renal disease. Then my resident took the genetic history and we drew up a pedigree. So if you take a step back, you can see everything you need to see. So that if you look at the generation 1, his grandparents, they're gone. If you look at generation 2, they are all gone, but we are able to start seeing something. This gene is running on the maternal side, purely. So that tells us it's a dominant, it's likely to be a dominant disorder because you don't need two genes to manifest the disease. That is one. But there are certain things I also want you to see. So if you look at the previous generation on his paternal unaffected side, there's a pretty heterogeneous cause of death, from diabetes, measles, drowning, hypertension, diabetes, and the like. If we come to the maternal side, it's consistent. Hypertension, CKD, CVA. So that is the phenotype. You know, something like polycystic kidney disease presents with CKD. They tend to present with hypertension as a result of the CKD. They tend to present with cerebrovascular accident. So we can begin to tell this family. So for a 61-year-old patient, we need to do surveillance for a cerebrovascular accident. If we look at this generation, we can see all of them passed on by the age of 70. If you look at the unaffected siblings, they live longer, so that we know the people who are getting this phenotype are dying by the age of 70. So now if we come to generation 3, which is where our index patient is, they are all in their 60s. Anybody who has the phenotype already has CKD. Hyperuricemia is a function of CKD. Our patient is actually coming for initiation of hemodialysis. His brother had CKD and got a kidney transplant. So we can begin to tell them, the people who develop this, who have this gene mutation, by the time they're in their 50s, they have progressed to end-stage renal disease. And if they are not able to get a transplant, they are likely to be deceased by the time they're in their late 60s or 70s. And the caveat is, if they're getting a living-related donor from the same side of the family, we need to make sure that the relative who's donating a kidney actually doesn't have the CKD, and ideally by genetic testing. So I'd like to highlight this as our index patient's brother. It was on the basis of him having CKD that his daughter was tested. She was not symptomatic, but now they did the genetic test and actually tested now essentially her daughter with a genetic test, so that you can map out and begin to advise this patient on what is expected or what the progression of this disease is likely to be, even in the asymptomatic persons. We know this is an autosomal dominant disorder, so we advise our patient that all his children need to be tested. If this was an X-linked, let's say our patient was carrying an X-linked recessive disorder, we'd tell him you don't need to bring, or even dominant, we'd tell him you don't need to bring your sons because you don't transmit your X chromosome to your son. So it helps you knowing the mode of inheritance helps you plan who to bring in for cascade testing and the like. So the advice we tell patients when we find a disease, genetic disorder, is that everybody in his maternal side needs to be tested. And we do what is called cascade testing, so that we have this brother who's diabetic. Maybe we test it, we find he doesn't have the gene mutation. We don't need to test his offspring, right? We come to the 65-year-old, we find they have the mutation, we test their offspring. So you test higher up as you can, where you don't find the mutation, lower down, you don't need to do. Okay. So how do you interpret a pedigree? It's usually so that, like, now you start looking for pairs. This is a mother to son, mother to daughter, and mothers transmit the same thing: autosome, X chromosome, mitochondria. If you have a father to daughter, which we have here, it's only autosome and X chromosome. So that rules out mitochondria. We know this is not mitochondria. This one doesn't show, this particular pedigree doesn't show a father, son, but if it did, it would rule out X, and that would leave us with an autosome. But we still have enough information because we know the phenotype is polycystic, it tends to be largely autosomal. So our conclusion just from the pedigree is that this is an autosomal dominant disorder. And remember, we said one gene causes the phenotype, so that we know the likelihood of passing on the mutated gene is 1 in 2. And actually, if we when we counted the number of relatives on this side, there were 25 and 30 of them had it. So it's actually obeying the principles of Mendelian inheritance, that the likelihood is 1 in 2, and it's a fully penetrant disorder because everybody who's having the genotype is actually manifesting the phenotype. And that is in keeping with what you know about polycystic kidney disease, that it's a highly penetrant disorder. Anybody with a genotype will develop the phenotype. Unlike the patient who started in where the child has autism and the father had the same gene and he was normal, so that one would say it's not fully penetrant. So another approach to a patient, so that like now we look at a syndromic approach. We have what we have is a sudden cardiac death in a 7-year-old. You can exclude, just for argument's sake, things like asthma and toxins. So you have what we think is a sudden cardiac death. You know, children should not be dying from cardiac disease. So now we start asking our question. This is likely to be genetic. So where do we start? Does a child have an inherited congenital heart disease? Down syndromes, 50% of them have some structural heart defects. Consanguineous unions tend to unmask rare autosomal recessive congenital heart disease. Then now we go structurally. Myocardium, is it hypertrophic cardiomyopathy, which is exclusively genetic? Is it dilated cardiomyopathy and restrictive cardiomyopathy, about half of which tends to be genetic? Is it arrhythmogenic, which can also be genetic? And then is it the pericardial diseases, which are genetic in origin? Is it a genetic coronary artery disease, as in familial hypercholesterolemia, which leads to extremely high LDL level? So that even with that, you're able to start thinking, okay, these are the tests. I will look for this gene mutation. If I'm thinking coronary artery disease, I'll look for markers for hypertrophic cardiomyopathy or dilated cardiomyopathy, which can even be at the phenotypic level in your ECG, your echo, or at the genotype level, which is your gene factor. So we try and narrow down what we are looking for, because genetic disorders are expensive. If we can try and narrow it down, the better. I mean, if I wanted to look for sickle cell disease, I can do whole genome sequence, look from chromosome 1 all the way to mitochondria, which would still tell me the patient has sickle cell disease, but then you've wasted a lot of money on a lot of non-essential. So we try and narrow it down. Where do you think this genetic defect is? And are there tests you can do to narrow down and save on cash? So now we go to genetic testing. After we do pedigree and we have an idea of how we want to do, we go to genetic testing. I've already alluded, they're extremely expensive and sometimes they don't give the answers. In fact, I've come to fear the day results are coming back, because after all this journey, you might come and find no mutation detected. Daktari, what can I do? We actually stop here. I can't answer whether you can transmit to the next generation because you don't even know what we are looking for. Maybe come see me in 2-3 years, maybe they'll have found a mutation that runs in your family. So it's a pretty dicey place to be. And then, so I don't always do genetic testing. Ideally, you want to do tests to confirm, but these are things that are not available to everybody. So that if a patient is coming with a congenital disorder, which I think is genetic, it will be prudent to do, like we said, intellectual disabilities, autism spectrum disorder, vis-a-vis teratogenic, which I might opt up to. And then we talked about disorders which are polygenetic, rare multisystemic disorders, early onset or atypical presentation. Alzheimer's is an interesting disease. When you're talking about late onset Alzheimer's, patients who are presenting beyond 65 years, 70, 80, the likelihood that that is being caused by genetic mutation that is known and identifiable tends to be pretty low, less than 1%. But the younger it appears, the more likely it is. So that in patients presenting under 65 years, 1 in 20 will most likely have a genetic mutation that you can map and try and look for the same in their offspring. So 1 in 20 is pretty low. So that test is not something I would prompt patients to go for unless they have very many relatives who have the early onset Alzheimer's. So you know this gene is pretty dominant in this family. There'll be a high yield of doing genetic tests. You'll be able to answer. And the more relatives who have the disorder, the more prudent it is to do testing. And first degree relatives and first degree relatives more, second, third. And someone says, yes, I had a great auntie who had Alzheimer's at age 50. Anybody else? No. That one is probably not somebody I will prioritize for genetic tests. But someone tells you their dad and their grandfather had. So you can tell it's pretty dominant in that. And then disorders where you're going to get a high yield. A disorder, so like diabetes, there's a monogenic form, maturity onset diabetes of the young, where one gene is the one that causes. There might be a yield. Type 2 diabetes, you'll do many tests, but you'll not be able to tell somebody whether they will develop the phenotype. You say, okay, your risk of, with these genes, these 50, 100 genes I've tested, your risk of developing type 2 diabetes is 2 to 3-fold higher than somebody who didn't have, which already could have told them, just looking at their pedigree, they have many first-degree relatives and second-degree relatives who are affected. That could have been earlier. And sometimes I opt out of genetic testing due to financial constraints. A disorder like trisomy 21 Down syndrome, the phenotype is pretty diagnostic. You might not prioritize genetic testing because a karyotype is what, 30, 40,000 shillings. Money that could be otherwise better spent in taking care of the child. So that one I would not do unless the parents are coming, they're asking what is the risk of having another child with Down syndrome. Now there we need evidence, we need to find out this, and trisomy 21 non-disjunction or translocation, so that you're working from a factual basis. But just managing the patient, if there are financial constraints, that one I would not prioritize. And in as much as possible, you want to do the gene mutation analysis on somebody with a phenotype. So this is, let's say you have two chaps, this lady and her son. The lady has the phenotype because she's shaded, right? Who would you do the genetic test on? Always, always, always as fast as possible aim for this. And why is that? Genetic tests, remember what we said about sometimes the mutation has not been characterized. So like this is a gene for cystic fibrosis. This up here is the cystic fibrosis. These are the introns and the extrons. From where my arrow is all the way down, these are the mutations. There are over 2,000 known, and the word is known. So that in a family, if you bring this lady and look for cystic fibrosis mutation, you look in all the 20,000 genes. If you don't get it, but she has the phenotype, yes, she has the phenotype, but your mutation has not been characterized. So we stop there. We can't even test the child because we don't know what the genotype is. But if you started with the son, if you don't find it, you don't know, is it because the child doesn't have the mutation of interest, or that the current tests that exist are not able to pick. So as far as possible, you want to look for somebody who has the phenotype to do. Same with a gene panel for hypertrophic cardiomyopathy. these are the genes that are associated. Somebody might come with classical symptoms, but you still miss it. So it means for that family, you're not able to tell them what you're looking for. And when we do genetic testing, there's a classification system. Benign, likely benign, which is where we want to be, or pathogenic. So it's most likely causing, likely pathogenic and pathogenic. The most troublesome to interpret is this variance of unknown significance, because you're saying I actually don't know whether it's okay or it's not okay. So it's a very dicey place to be. So how we do genetic testing, because when we do a genetic test, you might actually be discovering new mutations. So that you look at this, we said 6 or 7 genes that cause hypertrophic cardiomyopathy. And in one of them, you find a mutation that has not been characterized before. Okay. And all the others are normal. So then I'll list it as likely pathogenic, because in science, we don't base hard facts on just one case report. Most likely this is likely pathogenic. Maybe with time, as other people report the same, it will be upgraded to pathogenic, or it might even move to variant of unknown significance. Maybe it was a red herring. So this is a child with polydactyly, isolated polydactyly, no physical disabilities, normal neurological function. So is this a benign or pathogenic variant? It's actually a benign. And this underscores something I'd like to highlight in genetic disorders and in rare diseases. People look different. They are highly stigmatized. So that if something, this is actually, especially polydactyly, has been found to be a normal family trait, isolated. We're talking now about isolated polydactyly that runs in families, particularly Black families, tend to be autosomal recessive. There have been some dominance described. So that if this is, somebody comes with this, I'll probably not even do genetic testing. It's a family trait, it's isolated, and then we just take it at that. So underscoring is the importance of people look different, they become highly stigmatized. So we'll start looking at, so those are the genetic tests when you're looking at the genotype. Those are your C, T, G, and A's, right? So now we start looking at a macroscopic at the chromosomal level. And a karyotype essentially is cells that have been cultured and put in the prophase phase of cell replication. And they are looked at under a light microscope. We know the normal size and the size and G banding and the like. So you're able to tell we have a few things you're able to tell. We know that normal chromosome should be diploid. So we expect to, when you get to chromosome 7, you have a triploid, you have tetraploid in chromosome 8, and even the sizes are not uniform. So that if you come to chromosome, is this 13? Let me use this one, chromosome 12. We have different sizes. So how do you gauge whether this one is increased in size or this one is reduced in size? Remember, we said they're in decreasing size. So you compare with, it should be, so we know it should be a size between chromosome 11 and chromosome 13. So we know it's that this one has lost material. And we know that this is a male. So our karyotype, you should be able to tell the number of chromosomes at the very least and the sex. And sometimes you're able to tell whether you have a deleted material or increased. So like this one is a non-disjunction trisomy 21 in a female. This is a ring chromosome 15. So that if you look at this karyotype, you begin to ask yourself, okay, what will the phenotype look like? I'm not very sure. You go and look at the genes that are on chromosome 15, because those are the ones that are likely to be affected. And you try and read up on them and see where, what genes could be affected and how that can translate to the phenotype that you're observing. So this is a fish, fluorescence in situ hybridization. I like to call it a karyotype 2.0 because it tells you everything a karyotype tells you plus more. So that you're able to tell the name, size, and sex of a karyotype. But it gives you something else so that they color code, they fluoresce different, so that chromosome 1 has been color coded yellow. If you look at this fish, they actually look normal. So the karyotype for this might have been reported as normal, but there's quite a bit of translocation. It's picked genetic material from 7, 13 has picked from 19. So there's quite a bit of translocation. So fish is an upgrade from a karyotype. Karyotype, there are some things it's not able to answer. Right? We've already talked about genotyping, which is either looking at the DNA sequence of the patient you suspect to have a mutation against the known reference sequence. Then these are, I think I'll skip that. I want to mention other tests that are of interest. So this is what we call a two-parent paternity testing. So remember we talked about human beings are 99% identical. So when you're going to look at paternity testing, you go for the 0.1 in difference, right? Because if you go for the 99%, you might even pick the 60% that you share in banana so that this child's paternity is not known whether it's to be a human father or a banana. So you go for the hypervariable region. And we know everybody inherits a pair of chromosomes, one from mom, one from dad. And at a certain point, these are identical. This will have a certain nucleic acid sequence. So they are given numbers and names, but what it means, it's a series of C, T, G, and A's. C, T, A, G, C. So they are slightly different. So they are given different numbers. So this is the child. You know, one must have come from dad, one must have come from mom or a new mutation. So then this is a two-parent paternity test where the mother's maternity is not in question. So we can begin to exclude the mother and now generate what is called obligate paternal allele. So that at this level, the gene sequence, the child number was given 26 and 23. Mom has 26. So we know the child's 23 should come from dad. But then now when we look, it's not matching with the dad's. Dad's is 22 and 24, meaning this, we say that just numbers, there's a DNA sequence to it. Okay, so we go to the same one, same thing, those are not matching, but after that, everything matches with the child. So then they can use that to calculate what is called a paternity index and confirms this is 99.99. That is to account even for, unless the father has an identical twin, there is no other chance anybody else is likely to be the father. These two underscore the fact that mutations occur. So these were mutations in the child because this which number? 23 did not come either from mom or dad. So it's a new DNA sequence that is unique to the child. So it's a mutation. So this is what is called a motherless paternity test. For one reason or the other, the mother was unavailable for the test. Can you still infer something from it? Yes, you have the child's alleles and we have the father's alleles. Then we go down and look. We can't exclude the moms, but what happens when we get to this third one? Here means the child had 8 and 8. So we know an obligate, the father must have contributed 8 and 16. And we are still able to generate something and confirm that this is the child's actually father. He moved from an alleged father to confirmed. So I want to just touch a bit on reproductive genomics, just a bit to almost calculate. And there are various entry points for reproductive genetics. So you have your disorders of sexual development, either patients presenting with clinical Turner syndrome, primary ovarian failure, atypical genitalia, suspected genetic infertility and its causes, congenital abnormalities picked during ANC. So mom is fine, they find something not quite right with the baby, either through ultrasound, conventional non-invasive prenatal testing or genetic NIPT or invasive test. So basically, congenital abnormality that is fetus. A couple with an affected child contemplating a second pregnancy coming to you for genetic counseling and way forward. Mom, dad known to have a genetic disease or a disease that's runs in the family, and a fetus exposed to a teratogen and presenting with potentially congenital malformation. So there are various things in reproductive genomics. Will I be able to have a baby? if I have a child, will I pass this disease to my offspring? There's a lot of guilt and blame. What can I do to mitigate my risk in previous pregnancies? Is someone who'd had an affected child? Is the disease treatable or curable? Unfortunately, the answer this is almost universally that it's not curable. So when people are getting pregnant, they have all these grand plans, which come crashing down when they are told their child has either a confirmed congenital abnormality or potential, either during ANC or neonatal previous. And the themes are the same: profound shock, guilt, blame, and this is usually lifelong, running across various generations. But you'll find a grandmother who has a disorder their child has, their children have. So they are asking them, I have made my children and grandchildren unwell. And that self-blame and guilt never goes away. So it's our role to enforce that it's nobody's fault. We do not have a say which genes we inherit from our parents. We certainly don't have a say in which genes we transmit to the next generation. It's just a game of chance. So it's not anybody's fault. A lot, genetic disorders require a lot of compassion. One case I read about, I think it was a magazine article, it was a mother who had taken care of a child who was bedbound from birth, was approaching adolescence, and she went into panic mode. Periods are coming. I don't know how. On a child well taken care of, absolutely loved. So she approached a healthcare worker and asked, is it possible to have a hysterectomy? And she said it's okay to say it cannot be done or it can be done, but the response she got, she was almost feel like she doesn't love her child, yet she was looking for a way to almost simplify management of her patient because she felt at that point she could not cope. So there's quite a bit of compassion that is required. And what is our responsibility in reproductive genetics? You calculate risk for future pregnancy. So we do what is trio testing. If we find an affected child, we try and see, could it have come from the previous generation? Is it a new mutation? What is the likelihood of recurrence? So that for autosomal recessive traits, where both parents are carriers, there's a 1 in 4 chance. If it's autosomal dominant, 1 in 2. Down syndrome, the risk of getting Down syndrome in a baby at the age of 25 is 1 in 1,250. So it's pretty rare. But if she's had a child with Down syndrome, that goes up to 1 in 100. So having an affected sibling greatly increases the likelihood that you'll have another affected sibling, even if the parents are okay. Because there might be a pool of gametes, your ova and sperm, that carry that mutation. And despite all this intervention, there is no guarantee that the disorder might not recur. So you can do everything and there's still a recurrence. And then now we need to begin to have conversations with patients. So if our patient has sickle cell disease or is known to have their carrier, we need to screen their partners. One of my residents is looking at acceptability of premarital screening of sickle cell trait amongst people who are not yet married, so university students, because that is what is current. In the Middle East, where they have a lot of autosomal recessive inherited metabolic syndrome, they actually encourage people to get tested so that you're making informed decisions about coupling. If you end up with a partner who's carrying the same mutations, at least surveillance begins earlier, so that the diagnosis of sickle cell disease should be made immediately in the neonatal period, not when a child presents with their first sickling crisis at age 3, right? And ideally, one day we'll get there, where we're doing neonatal screening for inheritable disorders, which are prevalent in our community, so that the panel we'll be doing for neonates born in Kenya will not be the same panel you're doing for neonates born somewhere in Japan. It needs to be customized depending on the population studies that have shown, yes, these are the most common genetic disorders. So this is where we'll get the highest yield. So if we put our money here, we are likely to catch people earlier and intervene earlier. So there are times we opt not to do genetic testing. when you're doing trios testing, there's a likelihood that you might have unmasked previously misattributable parentage. And when you say parentage, you're likely talking about paternity. In medicine, genetics, like all other spheres of medicine that we usually just let it be. We do trio testing when we think we can answer the question, what is the likelihood that this will recur? How can I prevent a recurrence? But if a couple with a child with Down syndrome came to me to do trio testing to find out who's the contributor of the extra 21 chromosome, it tends to be maternal 80%, but up to 10-20% can be paternal. So you cannot know who was the contributor until you do. So that is not a tester will do, because it doesn't answer the likelihood, will I get another child? It doesn't have any medical benefit other than apportion below. Another time I might opt not to do a genetic test. So this is a 4-generation pedigree. You can see generation 4, 3, 2 are affected, and we have multiple affected in generation 2. So we know one of the persons in generation 1 carries the gene of interest, but they seem to be asymptomatic. Would I do a genetic test? They're in their 70s and 80s. So if they have not manifested the disease that is even presenting in people who are very young, it's unlikely to manifest. Will there be any benefit other than just making them feel guilty? It will have no clinical benefit. So there are times we opt out of genetic testing. So reproductive genetics, children born who have rare diseases who look different face a lot of stigma. I am not trying to, I'm just doing a comparison with 2 diseases. Children who have type 1 diabetes have to overcome tremendous obstacles, and I am not overlooking that. I just want us to do a comparison and kind of see what sometimes people go through when they have visible conditions that make them look different from others. So we have 2 children, one who has polydactyly and a child who is diagnosed with type 1 diabetes. So this girl will say, what is type 1 diabetes? Because she's explaining, even my shosho has, even my best friend's dad has, but his is not for Jidungai like mine, his is for swallowing. So that child will sort of have a community where they'll begin to understand, yes, I'm not the only one. This child may go through life without having ever met anybody who looks like them. So what happens when they go to school? The fact that somebody looks different will face a lot of stigma and be ostracized. So then they finish school. They start looking at their career choices. They've done pretty well. Both of them will become ineligible to join a military academy or flying school. The reason the diabetic will be told, we send people to boot camps in Abadea's forest for 3 weeks. You can't have such a rigorous training when you're insulin dependent. Okay? You can't join flying school because you can't risk you having hypoglycemic episode up there. Fine now. The child with polydactyly will also be told they're ineligible for those. Why? There is no reason, but they can't join, right? So then they get to medical school. Medical school, so long as you can meet the rigors of an medical school, you're good to go. But this child's father will have to make contact with somebody in the nursery area who makes gloves that fits this child. So they'll have to, when people are buying stethoscopes and books, this child will have to have extra money for gloves that will follow them for the rest of their life. So that when people look different or a rare disease, in fact, how do they say that insanity, you don't have to, it's just if you are the only one, you appear insane. So that rare diseases might not be pathogenic, but the fact that somebody looks different from the masses are already labeled to be at odds. So now we go to the mainstay of reproductive genetics. We are looking at a classical disorder of sexual development. So a girl comes with what looks like Turner syndrome. Turner syndrome, there's not much else that looks like Turner syndrome. So we know the phenotype is Turner syndrome. So what do you think the karyotype will be? Pretty straightforward, right? 45XO, because Turner syndrome tends to be an abnormal where you have only one X chromosome that is present. But that is not the end. They can be mosaics, so where they have populations of cells which are 45XO and 46XX. And the challenge comes in sometimes when you do a karyotype, whatever you're sampling from, whether it's a swab or blood, if for some reason you pick this one, they'll actually be reported back at 46XX. They're not done. You might go and find it's a karyotype 46XX. You do further tests, you find it's not mosaic. So they have 46XX, but there has been a submicroscopic deletion where of the short arm of chromosome X, where the genes for sexual determination lie. They're still not done. It can be 46XY. But what has happened to the Y, it has removed the genes that are associated with Y chromosome male feature. So like your sex determining region of Y chromosome. So what this underscores, when it comes to reproductive genomics, the first thing is to do a karyotype and try and see how far the genetic phenotype, the genotype, sex genotype deviates from the sex phenotype. Karyotype, 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 or whatever form. So that is the basis of that, right? And then I'll just sample something so that if you're looking at disorders of female development, if you go organ specific, ovaries require 2 X's. So that if you're beginning to talk about disorders of like Turner's, that might be the cause for ovarian dysgenesis. Now, if you look at the Müllerian system, it requires 2 things: absence of the Y chromosome and absence of your AMH hormone from the testes. So you expect the phenotype to be 46XX, but you can have 46XY with microdeletion. And then you go and try and find out, is it hypergonadotrophic, meaning the problem is at the gonadal level? Is it hypogonadotrophic, meaning the problem is at the hypothalamo-pituitary axis? And then there are genetic causes for those that you can evaluate. I want to spend a bit on time on external genitalia in a female, which is affected in visualization in congenital adrenal hyperplasia, which we'll come back to. And then there are some disorders that affect the sexual organ, but they also have other systems involved. Classically, one of these is your Fragile X syndrome, which is a X-linked dominant disorder, and it presents different. So that in males, the full mutation, they have your intellectual disability, ASDHD. Females with full mutation, mild asymptomatic, because they have 2 X's, so there is a dosage compensation. Then the males who have the pre-mutation, meaning it has not gone to the full mutation, they tend to be normally intellectually, but then they develop ataxia in their 5th, 6th decade. Females with pre-mutation present with premature ovarian failure. So that if you're dealing with a lady who's gone into menopause, everything was fine. She was doing fine. She got into her 30s and 40s and went into menopause. You need to begin to ask, other women in your family have undergone that? Is there signs of autism or ADHD in the male relatives? So that you begin to think, okay, maybe this is Fragile X syndrome. So when it comes to genetic evaluation of the reproductive system, we say it's a karyotype. So you do a karyotype. If it's normal, we start asking ourselves, it might be macroscopically normal, but let's look at the gene level. We see we have microdeletions of X chromosome or Y chromosome. If it's abnormal, you begin to characterize it. Okay. So when it comes to genetic evaluation in fertile men, okay, when you're looking at genetic evaluation in both males and females, especially people who are undergoing IVF and the like, by the time a lady is known to be menstruating okay, her hormone profile is okay, she's generating good quality of her, you pretty much know that there is nothing genetically that is abnormal. But if you're having a man who's having what looks like oligospermia or azoospermia, you need to start thinking about genetic disorders. Now, the caveat here is you might be able to do genetic tests to confirm the genetic mutation, but these people presented well in a fertility clinic. So they have spent 200-300 shillings, money they could have spent on another IVF cycle. You've just told them what they have. You have not told them how that one will change, will increase their likelihood in getting a baby. most of the time. Sometimes you're able to answer that, right? So then when you talk about male infertility, one of the disorders that comes up is Klinefelter's, which is 46XY. So they have lived their life normally. They have been okay. They are just noticed to have a reduced sperm count. If they undergo a successful IVF, they might pass on the extra X chromosome either to the female. This one will be asymptomatic, or they might develop another Klinefelter. So that these ones need probably the neonatal period to screen the baby for sex chromosome aneuploidies. So I'll just mention something about experimental genetics in recurrent pregnancy losses. There are male factors and female factors. Male factors, interestingly, so that somebody who looks normal, you've done your profiles, everything looks normal, healthy sperm count. But then when they start looking at markers that tell you DNA condensation, so you're looking at the DNA level, and this is just a protein marker. They found that this marker is highest in fertile. It was highest in men who are fertile, right? When they looked at the levels in men with recurrent pregnancy losses with normal sperm count, it matched those of infertile men. So that recurrently pregnancy losses or a repeated failed IVF procedures should sometimes prompt genetics testing in the male. I say sometimes because the question again, so you've done these things, it's not given me what I came to get from you. And now we are starting to have some infusions where they're trying to reduce the rates of fetal rejection in women who are having recurrent pregnancy losses. Prenatal testing and prenatal counseling is the most difficult aspect of genetic medicine. Because like we said, the child might have the genotype, but you're not able to tell that if this child developed a phenotype or not. If it's something that is known, like if you test a child in the neonatal period because the mother is known to have polycystic kidney disease and the child has, that one is straightforward. It's hard. They are guaranteed that they will get the phenotype. But this one, I don't know, wait and see. Something like hypertrophic cardiomyopathy, only 50% ever express the gene. But if you test 1 in 2, one will have a completely normal life, live to 80, 90, never get anything wrong with the myocardium or a conduction system. Another one will get an arrhythmia and die at the age of 24. And you're not able to tell who will get what. So genetics sometimes is, I can tell you the mutation, but I can't tell you what the future looks like. So that it's just a wait and see approach. Okay, so I'll just mention 2 or 3 things about teratogenicity. This is a study one of my residents who completed, and he looked at the residents' knowledge when it comes to teratogens, and correctly identified were less than 35%. So babies are exposed to teratogens, right? And it depends on the dose and time. So that in week 1 and 2, it's either all or none. If a fetus has a one-time insult like radiation, it might either lead to early fetal demise or they come out unscathed, right? And then now knowing where in the, knowing what develops when. So I want to look at something like congenital adrenal hyperplasia. This is a couple that has had a child who's previously affected. You did 2 year testing, so you know mom has the recessive trait, dad has the recessive trait. So there's a 1 in 4 chance of their fetus getting it. And we are worried about virilization in the female fetus because of excess testosterone. Now, fetal genital external genitalia is complete by week 9, right? We will not be able to tell whether this is a female fetus, whether they carry the mutation until week 10, right? So that in this, someone who's had a child with congenital adrenal hyperplasia, we start steroid therapy. Immediately they get pregnant to avert them getting a typical genitalia, which is more or less permanent, right? And then you can begin to drop the steroid because we don't want to expose unnecessarily. There's no, as much as possible, we try and remove drugs because steroids are not completely harmless, given at high doses and low. So at week 10, we can do a genetic NIPT, and if we find out it's a boy, we can drop it, right? Also at week 10, we can do the mutation so that if we find the child is not carrying the mutation, we can stop the steroids. So there's quite a bit. When you look at teratogenicity of fetal alcohol spectrum disorder, it affects both the mother who's consuming alcohol when she's pregnant and a father who's consuming a lot of alcohol, chronic alcohol consumption. And how the baby is affected by the alcohol is actually genetic. So that monozygotic twins who have identical DNA, concordance rate for fetal alcohol syndrome is 100%, approaching 100%. But when you talk about monozygotic twins, it goes down to 60%, meaning two dizygotic twins, you can have one who comes out completely unscathed and another one who gets very severe physical and intellectual illness. And it tends to affect the epigenetics, and it runs through generations. It's been shown in animal models, both rats and guinea pigs, so that if the father is a chronic alcohol consumer, generation 3 will still have behavioral issues, and it's largely tied to increased expression of your ACTH and high stress hormones. So almost to wrap up, genetic counseling, you're breaking down the phenotype, genotype, explaining inheritability, answering all the patient's questions, calculating risk, telling them who they need to bring in for testing based on the risk profile. And at the end of the day, the patient decides, give them all the information and make the decision on how they want to progress. So we are coming to the end. I want us to go back to this case that we began with, and we go line by line and answer. So we have clinicians at a university hospital. Clinicians at a university hospital first encountered the aggressive family when several women of childbearing age sought genetic counseling in 1978. So why are these women presenting to hospital? I'd like to open up, if I could get a few answers. Why are these women coming to hospital? Sentence 1. Why are the women of child? Yes, please.
Sorry, I was just saying they can raise their hands, then we can actually give them rights to speak if somebody wants to answer.
Or they can put in the chat box and read for me, just the first 2 or 3, so that I want us to think through, so that you have women of childbearing age who came for genetic counseling. Why are they coming? What is their concern? I'd like to see one of the overarching themes of genetic medicine. Has anybody attempted to answer, Dr. Amina?
So, male pattern aggression in family.
No, I first want to start line 1. We'll go line by line. So why is it that the women of childbearing age came for genetic counseling? What is the significance of that?
Pregnancy loss. Some people are, somebody's said pregnancy loss. Some people are saying male pattern aggression in family.
Okay.
Yeah. To have children, to see if they can have children.
So women of childbearing age, so they are worried. Will this disease pass to my offspring, right? That is one of the overarching themes in genetic medicine. Genes are inheritable, so can I pass this on? So now we go to the phenotype. Certain family members, always male, right? So we'll come back to that. The phenotype, mental retardation and aggression, right?
Yeah.
And then this phenotype was so alarming that in 1962, the uncle sat down and chatted and found that other 9 members are affected as far back. So it's 100 years, right? So why is it only male? When you start seeing only male, where is likely to be the location of that gene? And that tells us the most X. And what else?
Sex chromosome. And
X and which other one? Which one is unique to male?
Y.
So if it's X, is it likely to be dominant or recessive?
Dominant.
Recessive, because if it was
Recessive,
dominant,
sorry.
the women would also have, right? So we're starting to think this is definitely, because for 100 years, there's not been any mention of a
Women.
female, right? Yeah, so it's either X-linked recessive or Y-linked, right? And if it's sitting on the sex chromosomes, is it affecting genes of reproduction? We don't think so, because it has consistently existed for 100 years, right? So we've covered that. So after that, next thing, pedigree, always. So this is the pedigree of that family, and it showed, yes, it's an X-linked recessive disorder, so that it had affected males and carrier females. So it's an X-linked recessive disorder. The phenotype, I'm not sure what we are dealing with, but you know it sits on the X chromosome, and I really like X-linked disorders because it's narrowed down from 23 chromosomes down to 1. And if I pick an affected male who only has 1 X chromosome, I know it has to lie there. So that if I was to pick somebody to test, I wouldn't pick the carrier female because she has 1 normal X and 1 abnormal. So if I narrow it down to the affected male, he has 1 X chromosome, whatever is cooking in this family is sitting on that chromosome. So then we go on a hunting expedition. X chromosome has over 900 genes, and they have various functions. We know this is, so we start excluding. We know this is not hemophilia, this is not Alzheimer's, this is not Duchenne's muscular dystrophy, this is not retinitis. This is pretty much it. So now we go and look at the X-linked mental retardation syndromes that have been characterized. There are 23, but I'll narrow it down to 2, right? There's Fragile X, and there is the monoamine OA deficiency, right? Could this be Fragile X? We've seen it tends to show more of the intellectual challenges in the male. So we go back and ask, are the women reaching menopause in the 30s and 40s? Are there some mild subtle neurological symptoms in the females? No, so it's unlikely to be Fragile X because it's an X-linked dominant disorder. So from the 23, I've narrowed it down to Dr. Amina.
Yes.
Doctor, please mute your mic. I think it's still on. Thank you. So we have narrowed it down to maybe the monoamine A because we are seeing the phenotype is aggression and violent, but we're not very sure. So we start going into your genotype database and phenotype databases. So we go and look at what monoamine oxidase A enzyme does. It's associated, deficiencies in this gene is associated with Brunner syndrome and antisocial personality disorder. So now we go to a phenotype database. What is this Brunner syndrome? Yes, it tells us it's an X-linked recessive disorder, and it gives us the location. It's on the short arm of chromosome X. It's an X-linked recessive disorder, even the antisocial. So what is Brunner syndrome? The phenotype tells us it's a recessive X-linked disorder characterized by impulsive aggressiveness and mild mental retardation because of the deficiency, right? And the reason, and this deficiency is most prominent in the frontal lobe, so they have poor impulse control, and the limbic system, so they have, what does it do? Sorry. Poor emotional regulation, right? And the name Brunner syndrome actually came from that 1978 family. So then we have a diagnosis. And then if you're going to, what they did that time, they measured either the metabolites, the monoamine A metabolites, or the enzyme, right? But now you're able to test the gene level. So genetic testing can come at any level. So moving from this to, I don't know what we are dealing with, to this, just requires you to have a systematic way of doing it, do quite a bit of reading and come out with it. So that for this family, I would just probably go and do the monoamine A, the gene level, mRNA level, the protein level, or metabolite level. And that is the end of my presentation. Thank you very much for your attention. Maybe I hand it over to Dr. Amina.
Yeah, thank you too. The end was was quite interesting. So just to make sure I got you right, Dr. Ray, you said the X chromosome has about 900 genes, which out of the 900, you'll have 23 which are linked to mentally retarded
Syndromes.
disease, but I don't understand how you broke it down to 2 of them out of the 23. That part I got lost.
You go step by step. You go,
Okay.
the genotype, phenotype, they pass this read. This doesn't look like what I'm dealing with. This doesn't look. So it's actually go. Sometimes it might take quite a bit of time. I just broke it down for simplicity. But now we know our gene sits on chromosome X, right? And there are 23 characterized gene mutation. So you just go through all of them and reading both the gene and the phenotype and trying to see where to narrow it down.
If
that fails, you can do sequencing of the whole chromosome and hope you pick something so that you could, if this wasn't fitting in, you might go and do this and then go and find actually the deficiencies in this enzyme. Then you go back and read. But that is an expensive because you've gone and spent money doing 900 genes, including for hemophilia, Duchenne's.
Yeah.
Right?
Doesn't make sense.
Yes. So that you try and break it down and say, okay, so what might I be dealing with? How do I prioritize testing? Because testing is expensive.
So in a nutshell, that family had Brunner syndrome. Did they test? We'd like to know the ending.
Yes.
It was tested and confirmed.
Yes, actually the name Brunner syndrome came from that family. And in 1993, they didn't have genetic tests. So they tested the metabolite of the enzyme. Remember when we said you can test at the gene sequence level, the mRNA sequence level, the protein level, its metabolite, so that even something like sickle cell, if you look at your R disease under a microscope, you can begin to tell you, I think there's sickle cell here, right? So you can even test at the phenotype level. So they tested either the enzyme, which is the protein, or the expected metabolites, which are biochemical tests. I forget which one of the two it was. So the name Brunner syndrome came either from the name of this family or the doctor who described it in that pedigree in 1993.
Wow, that's quite interesting information.
Okay.
Thank you very much, Dr. Ari. You have really engaged us and you've really generated a lot of discussions from the audience. I hope we'll be able to answer some, if not all of them. So first question was from Professor Lema. Are we able to do genetic manipulations here in Kenya? If not, are there plans to do that to address genetic disorders?
The short answer is no, but we believe good things are coming. It's possible, it's being done, right? So that's a disorder like sickle cell disease. They're actually doing, like to just use the words, they're actually doing gene editing and doing stem cell transplantation with a hope to cure and get now somebody producing. Because they'll still have to take lifelong immune suppressant. But sickle cell disease is chaotic. We've all seen it, right?
Yeah.
Okay, someone has asked, can blood transfusion change genes in any way? No, they're not incorporated into the system because those ones are cleared, right? They don't form part of you. The RBCs are existing in circulation, but within a certain amount of time, they have been cleared. The only thing now is that you don't want to do genetic tests at somebody who's had a transfusion because you might pick, are you calling them the transfusion? The donor DNA test from the white blood cells, not the red blood cells. Red blood cells don't have DNA.
Okay. The other questions, there are two of them. Yeah.
Suppose I don't have anybody to give a true history concerning family genetics. Is there a way to move forward? Yes, so that if somebody comes in who's been adopted into a family, family history is unknown. There are still ways to go around it, but then that means you'll have to cast a wider net so that if they come with a typical presentation, like that Brunner's family, if you just had one person coming in, adopted into a family, so he's the only one, we'd have to do whole genome sequencing. We look at all 23 chromosomes, mitochondrial, and look at the ones that give us abnormal gene mutations. Can any of these mutations explain what is going on, right? So it's the same way if a patient comes with loss of consciousness, you will still know how to proceed. It will just be a long way to go about it, right? Of course, I've talked about. Okay, let me read Catherine Miners.
Let me pick some random ones from the ones that have already been asked to make you a bit easier and to make it a bit more engaging. So there's a question from Gingo. He's asking, in terms of reproductive genetics, any information about polycystic ovarian syndrome, is it quite common? Because we talked about the common genetic diseases and the rare ones. And also in line with that, because it's still on reproductive, probably you can also expound, is there any role of genetics in preeclampsia in women?
Yes. Okay, let me start with preeclampsia. There are certain markers that have been found to be increased in preeclampsia. First, how do I begin? Your genes play a large role. In some families, it might be a stronger role. When you ask history and find someone has multiple relatives affected and very close first degree relatives, that makes the likelihood that genes are playing a bigger role, right? A disease like PCOS falls almost in the spectrum of metabolic disorders, loosely, right? So that, and those have genes associated with it. So when you're saying a disease might have a genetic contribution, there are others that have environmental. For simplicity of this presentation, I was just looking at genetics. So even when I was talking about diabetes, I wasn't looking at the environment so that we handle the genetics. There are a lot of things that come into play. So that something like PCOS, you will not do genetic testing. They will not change. They will not change anything, right? So someone has asked a very interesting question. The reference genome is an agreed upon standard, no race preference. Actually, that is not correct. So that, like that NCBI database, the normal reference sequence came from somebody. So they looked at, let's say 100 people, right? And looked at, when there were 100 healthy people, and they looked at their insulin gene sequence. And then they agreed, okay, it seems to be like this. But you know, out of those 100 people, none of them was a Kenyan from the Kano Plains, right? They tend to be Eurocentric. So that with time, and it will come to happen, as we start doing population genetics in our people, we'll start using, so that if I'm looking at your insulin gene and you're a Kenyan, I should be using a reference gene from a Kenyan. And then not all Kenyans are equal. Somebody who lives in the highlands, their gene are not identical to somebody who lives somewhere in the lowlands. And how do we know that? We all look different. So if we look different, even inside we are different. So the future of genetics is that we will come down to, as much as possible, doing reference sequence with somebody who looks like it, because then they become more informative. If I'm using somebody who's Scandinavian, I might pick what looks like a mutation in a Kenyan, where in actual fact, that is what our normal sequence should be, right? So the interpretation of that, or because they have found the mutations of polycystic kidney disease to be these ones in this part of the world, the fact that ours have not been characterized, I may do as someone who comes with that phenotype. I know there's nothing else, but then you don't get it. So it means maybe we have a genotype that has not been yet picked out. Maybe one more.
Yeah, so really now I just wanted to understand, Dr. Ari, because you say this field is evolving. There's a lot of talk on precision medicine, and I wanted to understand the role of a clinical geneticist in terms of precision medicine.
Genetics is so wide. So I left out oncogenetics and I left out precision medicine, pharmacogenetics, right?
Okay.
So me and you have different genes. Let's just take like cytochrome P450, right? Which is an enzyme, it's a protein. So it's encoded for by a gene. So my cytochrome P450 cohort of proteins will act differently compared to. So that let's take a drug like azathioprine. There's a certain gene, it's called TPMT gene, that if it's reduced or absent, predisposes people getting azathioprine to getting myelosuppression, very bad toxicity. So ideally, before you start somebody on azathioprine, now we're talking about oncology and our rheumatology and nephrology. Ideally, they should have that test done so you decide who not to do. The cost of genetic tests will come down. right? In the next few years, decades, so that we'll have almost like a gene passport, right? So that you've mapped out my mutated genes, you've mapped out my enzyme metabolism gene, so that you look at it and say, no, I can't give you as a therapy. I can give you this hypertension because this antihypertensive because this gene works. So that is the future. It's not too far off. Once genetic tests become cheaper, and things get cheaper with time, they will become the reality.
Okay. So there was a question actually regarding where people can find you in case they need referrals. So Dr. Sokao works in University of Nairobi. She's a lecturer, but she also has a private practice in Fifth Avenue, Gong. I think you can get more information if you'd ask our email. And more or less, Daktari, in summary, what's your parting shot for everybody? Because we've really learned a lot, and I think a lot of us will go and listen to the YouTube again. So, but we would like you to summarize it maybe in a minute or two.
Okay.
What's your take-home message for us?
Okay. So thank you so much, Dr. Amina. But I want to ask you the question. I want to throw back the question that you asked me. Are genetic disorders common? After this presentation, what is your take?
Yes, they are very common.
They are very common. We need to look for them. And when you start taking a genetic history pedigree, genetic counseling, and it just takes, once you understand what you're dealing with, is when you begin to answer. So that if I'm a male who comes with hemophilia, you know hemophilia is an X-linked recessive disorder, right? And they are pregnant, and you do a fetal scan that shows you it's a boy, right? So what do you tell them? What is the likelihood that that boy will get hemophilia? 0, because the father did not contribute any X chromosome. So sometimes it just takes understanding modes of inheritance and the phenotype and beginning to give patients direction, because after that, then they ask, okay, that child can't get. Do I need to test? No. Basically, they have X, it's on X, you have X, you did not give your son X. It's okay, they will never get. So there's just that assurance, so that child can exit the genetic clinic. You understand? So sometimes it's just understanding and being able to begin to guide. The cornerstone of genetic medicine is genetic counseling, and it takes various forms.
Okay. So thank you very much, Dr. and thank you everybody for listening in. For this particular session, we will upload the session on our YouTube channel. We'd like to say good night and kwaherini. Asanteni sana.
Okay, thank you very much.
Okay, bye-bye.
Yeah.